All right, let's dive in. This research on AlphaFold-3, this AI, it's predicting the 3D shapes of biomolecules. We're talking proteins, DNA, like everything.
It really is fascinating. It's not limited to just one type of molecule or interaction. AlphaFold-3 can handle protein complexes, interactions with DNA, RNA, even small molecules and modifications. It's a real game changer when it comes to scope.
Okay, so this deep dive is about more than just AI is cool. We're getting into how AlphaFold-3 actually works and what makes it so different, why this matters for, well, everything. We're going deep into like the nitty-gritty of this research.
Absolutely. One of the key things to understand about AlphaFold-3 is its accuracy. It consistently outperforms other models, previous models, and those specialized tools, often by a significant margin. It's like comparing a skilled craftsman with a whole team of robots. The robots can do a lot, but the craftsman brings that extra level of precision and artistry.
Okay, I like where you're going with this. But before we get ahead of ourselves, let's break down how AlphaFold-3 actually achieves this accuracy. This research paper mentions a shift from evoformer to pairformer for processing multiple sequence alignments. Can you unpack that for us?
Sure. Think of it like this. AlphaFold-2 relied heavily on analyzing the evolutionary history of a protein sequence to understand its structure. It's like trying to understand a person by looking at their whole family tree. AlphaFold-3, on the other hand, focuses more on the relationships between the individual building blocks of the molecule itself, like the amino acids in a protein. It's more like understanding a person by, observing their individual personality traits and how they interact with others.
So instead of just relying on evolutionary history, AlphaFold-3 is able to see the bigger picture. It focuses on the specific relationships, right, within the molecule itself. That's cool.
It is. And the shift allows AlphaFold-3 to be much more efficient and accurate, especially when dealing with complex structures or interactions that haven't been well documented in those evolutionary databases.
The paper also highlights the use of a diffusion module in AlphaFold-3. Can you explain what that is and how it contributes to the model's accuracy?
Imagine you have a drop of ink spreading in water. That's diffusion. Now imagine reversing that process, starting with the blurry, spread out ink and gradually condensing it back into that precise drop. That's what the diffusion module does with protein structures. It starts with a noisy representation of the structure and gradually refines it until it achieves a clear, high-resolution model.
So it's like taking a blurry picture and slowly bringing it into focus. That makes sense. But how does AlphaFold-3 know when it has achieved the correct structure? How does it, avoid getting stuck in that blurry state?
That's where the confidence measures come in. AlphaFold-3 uses a combination of these metrics, PLDDT, PAE, and PDE, to assess the accuracy of his predictions. Think of these metrics as internal fact checkers. They help AlphaFold-3 identify areas of the structure where it might be less confident and flag those areas for further refinement.
Okay, so it's not just, blindly spitting out a structure. It's actually evaluating its own work and highlighting potential issues. It's pretty sophisticated.
It is, and these confidence measures are so incredibly valuable for researchers because they provide a way to assess the reliability of the predictions and focus their attention on areas that might require further investigation.
So by combining this new pairformer approach with a diffusion module and built-in fact checkers, AlphaFold-3 achieves a level of accuracy that really surpasses previous models and those specialized tools. That's pretty impressive.
It is, and it's important to remember that AlphaFold-3 is, it's a generalist. It can handle a wide range of biomolecules and interactions, unlike some of those previous tools, that were often specialized for specific tasks.
Right. It's like having a Swiss Army knife for structural biology,
Right.
one tool that can do it all. But even the most versatile tool has its limitations. Let's dive into some of the challenges that AlphaFold-3 still faces.
Of course. While AlphaFold-3 is remarkably accurate, it's not perfect. It can still make mistakes, particularly when dealing with certain types of structures or interactions.
For instance, the research paper mentions the possibility of stereochemical errors. Can you explain what those are and why they matter?
Think of it like building a Lego model. If you accidentally put a left-handed brick where a right-handed one should go, the structure might look okay, from a distance, but it won't be quite right. That's a stereochemical error. It's a subtle but important mistake that can affect the molecule's function.
So even though the overall structure might be correct, these small errors in chirality, the handedness of the molecule could have big implications for how how it interacts with other molecules.
Exactly. And AlphaFold 3, like any computational model, can sometimes, sometimes make these kinds of mistakes.
The research also mentions challenges with predicting structures in what they call disordered regions. Can you shed some light on what those are and why they pose a problem for AlphaFold-3?
Imagine a protein as a chain of beads. Some parts of the chain might be tightly coiled, while others might be loose and floppy. Those loose, floppy parts, those are the disordered regions. They don't have a well-defined 3D structure, and they can be difficult to predict because they're constantly moving and changing shape.
So it's like trying to predict the shape of a string blowing in the wind. It's like inherently difficult because it's not static.
Exactly. And AlphaFold 3, which is trained on data from proteins with those well-defined structures, can sometimes struggle with these disordered regions. It might try to impose a more rigid structure on them than what actually exists in reality.
That makes sense. So it's important to remember that AlphaFold-3 is a powerful tool, but it's not a magic bullet. It has its limitations, and researchers need to be aware of those limitations when interpreting its predictions.
Absolutely. And with that in mind, let's move on to some of the more successful predictions and see what AlphaFold 3 can do when it's firing on all cylinders.
Oh, sounds good to me. Let's see what this AI can really do.
One of the most impressive examples from this research is the prediction of the human 40S small ribosomal subunit. This is a massive, massive complex of proteins and RNA that plays a critical role in protein synthesis, essentially translating genetic code into the building blocks of life.
Wow, that sounds incredibly complex. I remember from like biology class that ribosomes are like the protein factories of the cell. How on earth did AlphaFold-3 manage to predict the structure of something so intricate?
It's truly remarkable. We're talking about a structure with over 7,000 residues. Those are the individual building blocks of proteins and RNA. AlphaFold-3 not only predicted the structure of the protein components, but also the RNA component, known as 18S ribosomal RNA, and it even captured the tRNA molecule, which is involved in initiating protein synthesis.
So it's not just predicting the individual pieces, but also how they all fit together in this elaborate molecular machine that's mind-boggling.
Precisely. And this ability to predict the structure of these large multi-component assemblies is one of the things that really sets AlphaFold-3 apart. It's like having a 3D model of a whole factory, not just the individual machines within it.
That brings us to another area where AlphaFold-3 has shown significant improvement, predicting antibody-antigen complexes. The paper specifically highlights an example with the monoclonal antibody 15B6 bound to a mesothelin peptide.
Yes, this is a fantastic example of how AlphaFold-3 can be used to understand the interactions between antibodies and their targets. Mesothelin is a protein often found on the surface of certain cancer cells, making it a potential target for those antibody-based therapies.
So by being able to predict the precise structure of how this antibody binds to the mesothelin protein, researchers could potentially design even more effective therapies.
Exactly. It's like having a detailed map of the battlefield.
Yeah.
By understanding exactly how the antibody and antigen interact, researchers can engineer antibodies with higher affinity and specificity, leading to more potent and targeted treatments with fewer side effects.
That's really promising. But I also remember the research mentioning some caveats with these types of predictions. Didn't they say something about needing a lot of computational power?
Yes, that's right. While AlphaFold-3 is, incredibly powerful, predicting those antibody-antigen interactions can be computationally expensive. To achieve the highest accuracy, it often needs to, generate multiple predictions and rank them based on confidence. This is because the interactions between antibodies and antigens can be, they can be very complex with subtle variations in binding sites and orientations.
So it's not a simple matter of just plugging in the sequences and getting a perfect answer. Researchers need to be aware of the computational demands and the potential need for multiple runs to ensure they're getting the most accurate predictions.
Precisely. It highlights the fact that AlphaFold-3 is, it's a tool, and like any tool, it needs to be used thoughtfully, with an understanding of its strengths and limitations. Now, while we've seen some of the remarkable things that AlphaFold-3 can do, it's also important to, to be aware of the areas where it can still stumble.
Okay, so let's talk about those limitations. You mentioned stereochemical errors earlier. Can you give us a specific example from the research to illustrate what those look like in practice?
Certainly. The paper shows an example with the prediction of thermatoga maritima I-glucuronidase bound to beta-D-glucuronic acid. This is an enzyme involved in breaking down carbohydrates.
Ah, enzymes, those biological catalysts that speed up chemical reactions in living organisms. I vaguely remember them from high school biology. But what went wrong in this prediction?
Well, in this case, AlphaFold-3 predicted the incorrect chirality for the glucuronic acid molecule. It essentially flipped the orientation of a key chemical group. To use a simple analogy, imagine trying to put your right hand into a left-handed glove. It just won't fit properly.
So even though the overall structure of the enzyme might look correct, this small error in chirality could throw off the entire interaction.
Exactly. This tiny error can significantly affect the molecule's ability to, to bind and interact with the enzyme, potentially leading to, incorrect interpretations of the results.
So attention to detail is crucial even at the atomic level. What about those hallucinations we talked about earlier? Can you give us a real-world example of that limitation?
The paper shows a fascinating case with the nuclear pore complex. These are massive protein assemblies embedded within the membrane of a cell's nucleus, acting like gatekeepers that control what goes in and out.
So they're essential for the cell's function. They're controlling the flow of information and molecules between the nucleus and the cytoplasm.
Exactly. The specific nuclear pore complex in the study had a large number of unresolved residues, meaning their exact positions weren't clear from the experimental data. Think of it like having a puzzle with missing pieces.
And AlphaFold-3 tried to fill in those missing pieces.
Precisely. And while it did a decent job of predicting the overall structure, it ended up creating a more ordered structure in those unresolved regions than what likely exists in reality. It's like trying to force the wrong puzzle pieces into the gaps,
All right.
creating a distorted picture.
So it's not always about AlphaFold-3, getting things completely wrong, but rather sometimes it might overcompensate for missing information, leading to a slightly inaccurate representation.
You've got it. This tendency to hallucinate structure can be particularly tricky in those disordered regions, where the lack of a well-defined structure is actually important for the protein's function.
Okay, so we've got stereochemical errors where details matter at the atomic level, and hallucinations where AlphaFold-3 might get a bit too creative with filling in the missing information. Are there any other limitations worth highlighting before we move on?
The research also mentions that AlphaFold-3 can sometimes struggle with predicting the correct conformational state of a protein. Remember, proteins are dynamic molecules that can change shape depending on their environment and interactions.
So it's not just about getting the basic 3D structure right, but also understanding how that structure might shift and change under different conditions.
Precisely. For example, the paper describes the prediction of cereblon. This is a protein involved in regulating protein degradation. Cereblon can exist in both an open and closed conformation, kind of like a clam opening and closing its shell. However, AlphaFold-3 consistently predicted the closed conformation, even in cases where the open conformation was more likely.
So it's like AlphaFold-3 took a snapshot of the protein in one specific state, but missed the bigger picture of how that state might change dynamically.
Exactly. And this highlights the importance of considering the biological context when interpreting AlphaFold-3's predictions. It's not always about finding a single correct structure, but
Yeah.
rather understanding the range of possible structures and how they relate to the protein's function.
That's a really important point. So while AlphaFold-3 is, it's a powerful tool, it's not a replacement for critical thinking and biological understanding. It's a tool that can provide valuable insights, but those insights need to be interpreted within the broader context of the biological system being studied.
Absolutely. It's like having a high-powered microscope. It allows you to see things that you couldn't see before, but you still need to know, what you're looking at
Yeah.
and how to interpret the images.
So with that in mind, let's shift gears and talk about the bigger picture. What does AlphaFold 3 mean for the future of structural biology? What are the potential implications of this technology, both in the scientific world and beyond?
Well, on one hand, it's a major leap forward in our ability to understand the molecular world. AlphaFold 3 is like having a Rosetta Stone for proteins, allowing us to decipher their structures and unlock the secrets of their functions.
It sounds like we're entering a new era of discovery,
Yeah.
where the 3D structures of proteins are no longer, a mystery, but a readily accessible source of information. What are some of the key takeaways for someone who might be, encountering this technology for the first time? It's like we've been handed this powerful new lens, for exploring the molecular world.
Exactly. And this new level of understanding, it has the potential to revolutionize so many different fields. One area that's particularly exciting is drug discovery.
So how does AlphaFold 3 change the game when it comes to developing these new drugs?
Traditionally, designing new drugs has been a bit like trying to fit a key into a lock without knowing what the lock looks like. You have to try lots of different keys, hoping to find one that works. But with AlphaFold 3, we can actually see the shape of the lock, the protein target in exquisite detail.
So instead of fumbling around in the dark, researchers can now design drugs that fit perfectly into the active site of that target protein.
Precisely. This level of precision can lead to, more effective drugs with fewer side effects. It can also significantly speed up the drug discovery process,
Wow.
potentially saving years of research and development.
That's incredible. It sounds like AlphaFold 3 could be a real game changer for treating diseases like, cancer, Alzheimer's, and even those caused by emerging viruses.
Absolutely. And it's not just about developing these new drugs. AlphaFold 3 can also help us understand how existing drugs work, potentially leading to new uses for those drugs or improvements in their effectiveness.
So it's like having this molecular detective on the case, helping us understand how drugs interact with their targets and why they work or don't work in certain cases.
Exactly. And beyond drug discovery, AlphaFold 3 has the potential to revolutionize other fields as well. For example, in agriculture, it could be used to design more efficient enzymes for biofuel production, or to engineer crops that are resistant to those pests and diseases.
So it's not just about human health. It could have a major impact on sustainability and food security as well.
Precisely. And these are just a few examples. The potential applications of AlphaFold 3 are vast and still being explored. It's a truly transformative technology that could change the world in countless ways.
It's inspiring to think about the possibilities, but with great power comes great responsibility.
Yeah.
Right? What are some of the ethical considerations that we need to keep in mind as this technology advances?
That's a crucial point. As with any powerful technology, it's essential to use AlphaFold 3 responsibly and ethically. We need to make sure that it's not used for those malicious purposes,
Yeah.
such as designing harmful biological agents or creating new weapons.
So open access and responsible development are key to ensuring that this technology is used for good.
Absolutely. We need to have open discussions about the potential risks and benefits of AlphaFold 3 and establish clear guidelines for its use. We also need to make sure that the benefits of this technology are shared equitably so that everyone can benefit from the advancements it brings.
Well, we've covered a lot of ground in this deep dive, from the technical details of AlphaFold 3 to the profound implications of its capabilities. What are some key takeaways for our listener as they continue to explore this fascinating world?
I think the most important takeaway is that AlphaFold 3 is a game changer for our understanding of life at the molecular level. It's a tool that has the the potential to revolutionize so many different fields, from medicine and agriculture to environmental science and beyond. But it's crucial to remember that it's just a tool, and like any tool, it needs to be used responsibly and ethically.
So embrace the possibilities, but proceed with caution and a critical eye.
Always good advice. And to our listener, thank you for joining us on this deep dive into the world of AlphaFold 3. We hope you found it informative and thought-provoking.
Keep exploring, keep learning, and keep asking questions.
Yeah.